Now we are coming to animal cloning. Okay, we are coming to the next one, animal cloning. Now what is the meaning of cloning? Developing the genetically identical individuals is called a cloning. Okay, if the genetically identical animals are produced, then it is called animal cloning. I'll tell one example, simple example for this. One cow we are taking. This cow is having 1000 characters. In the 1000 characters, 90% of the characters are excellent characters. Good. So the animal is very strong. Okay, the milk yield, everything is very strong. Okay, such animal we want the same copies many. Okay, but if we allow them to reproduce natural method, some of the qualities may be lost, but we don't want. The 90% of good quality should be maintained. For that, cloning technology is very, very useful. Now by cloning, we can produce enormous number of the cow, but all are having same genes, 90% perfect and strong genes. That is 90% of the characters will be stronger. That is called a cloning, animal cloning. Production of genetic wise identical individuals, either naturally, we can do by nature method, and also by artificially. That is called a cloning. Natural clones are made by asexual reproduction. There are certain lower organisms, animals, they are reproducing by asexual method. No reproductive organs, no male animals, no female animals and all. I'll give one simple example. Amoeba, unicellular organism. In the amoeba, you can't identify male amoeba, female amoeba. No such segregation. Well, they are very common. One amoeba divides into two by binary fission. It is called asexual reproduction. So when one organism is divided into two, size will be small, but the characters 100% will be the same. That is called natural clones. The two organisms. Again the two they are growing. Again divided into 2 into 4. All the 4 will be clones. Genetically they are identical. So any organism which are reproducing by asexual method are always producing natural clones. First artificial clone. Okay, animal clone, first artificial clone, Dolly. It is a sheep. Sheep Dolly was developed by Ian Wilmot and Campbell in 1997. They only first developed a sheep Dolly, very, very pet animal. Okay, that is a cloned animal they developed now first in the world. Okay, how the sheep Dolly was developed by Ian Wilmot and Campbell, we have to see. Now, not only two scientists, they are the head of the research, but other scientists were also helping. Now, here we are going to take first one sheep. I am going to take, it is a female sheep. Okay, it is a female sheep. This female sheep is having excellent qualities. Okay, the female sheep is having excellent quality, good quality. 90%, why 90? 99% qualities are excellent. Okay, imagine like this. But we want the same copies many. We want the same copies many. What to do for that? Now what we have to do is, from the animal's body, one body cell should be taken. What Ian Wilmot did, this is a female sheep. Okay, this is a female sheep. It is having udder, U-D-D-E-R, U-D-D-E-R, udder. Udder means, now see that, udder. Udder means it is a mammary gland, milk producing gland. It is present here. From the udder, they have taken one body cell. Now this is one body cell, it is having one nucleus, and in the nucleus chromosomes are there. So it is one body cell that is separated. But they are not killing the animal at all. Just they are taking one group of cells, one cell is isolated. Okay. Now this animal is living. Now from this cell, they are taking the nucleus alone. They are taking the nucleus alone. Okay, that is called enucleation. Removal of nucleus is called what? Nucleus, it is called enucleation. Why they have removed only the nucleus? Because only the nucleus is having chromosome. Inside the chromosome only genetic material is there. That is what? DNA. So they have taken only the nucleus. So cytoplasm, plasma membrane, all these things are not important. They are taking only the nucleus. Okay. Actually, they have not separated the nucleus. The whole cell they have taken. They preserved the cell. Okay. The nucleus is also there. Then they have taken another one female sheep in the same group. Female sheep, another one. But it is not having that much of good quality. Okay. They are not having that much of good quality. But the sheep is producing one ovum. It is producing one ovum. Otherwise called a what? Egg. Okay. It is a bee. Okay, the sheep B. This is sheep A. This is sheep B. This is female. This is also female. Now we are taking one ovum. Now the ovum is having a nucleus. But in the nucleus, we cannot say that the chromosomes are having all good quality genes. So what we have to do? We have to remove the nucleus from this. Say this cell is having nucleus with the good quality genes, but here the nucleus is removed. I already told you, removal of nucleus is called what is that? Enucleation. Once the nucleus is removed, what is this? This is the ovum without nucleus. It is a ovum without nucleus. It is having plasma membrane, cytoplasm is there, but no nucleus. Now what they are doing? They are allowing the body cell with a nucleus, and they are allowing this ovum without nucleus to fuse with each other. Fusion of the two cells. Cell membrane and cell membrane join together, becoming a larger cell. Cytoplasm and cytoplasm will mix with each other because they are gel-like substance. Easily they can mix. So the mixture of cytoplasm will be there, but nucleus alone will be there. Nucleus of the first sheep that is present here. Okay, this cell is a hybrid cell. Okay, cell is hybrid, but chromosomes are same. Now this cell is entering into the culture medium. Okay, it is culture medium, a liquid, where the cell is keep on multiplying, enormous number. 1 cell into 2, 2 into 4, 4 into 8, like that keep on doubling. Then slowly, under suitable temperature in the laboratory, all the cells join together. Enormous number of cells are produced. They join together and form a blastocyst. Already I told you blastocyst. It is also called a blastula. It is a hollow spherical ball-like structure with the cells at the peripheral region, a wide cavity in the center. It is called a blastocyst. Once the blastocyst is formed in the laboratory in a petri dish or in a tray, then this blastocyst is introduced into the third female sheep. Only female sheep are taken. Now the third female sheep is having very strong uterus. Uterus is very, very strong. Okay, so this animal is going after giving one cell. This animal is going after giving one egg. But this animal is taken, it is having very strong uterus. So in the uterus of the third female, they are introducing this blastocyst. Then it is attached with the wall of the uterus, started growing. Then finally, after the pregnancy of 6 months, then the young one is developed, the fetus is developed, and this third female is delivering a baby, a young one. Okay, this young one is a young sheep. They gave the name. This is a cloned animal. This is the dolly name. Now they wanted to know whether the dolly is having the characters of this mother or the second one or the first one. They observed that this sheep dolly is having all the characters like the first sheep, because this is the sheep which contributed the genetic material in the nucleus. So the same characters are present. So this is the clone of this animal. It is not having any characters like this female which gave the egg, this female which was bearing the growing fetus. Okay, now it is having. So this is the clone of the first sheep. Because I already told you, this sheep is having excellent qualities, even the sheep dolly was having. This is the first clone what was developed among the mammals. Okay, not other animals at all. Among the mammals, it was a pet animal like dolly they gave. But this dolly was living only for 10 days, 11th day it died. Okay, that is different. But how the cloned animal is produced? Like that, any number of, from one cell we can produce one clone. So like that, any number of cells we can take from the animal, we can produce any number of copies, genetically identical animals. Clones can be developed. Now these are the steps involved into that. Now we are coming to advantages of animal cloning. Actually advantages are there, even disadvantages are also present. Actually disadvantages are more, but advantages, we will take that. The advantages, the cloning offers benefits for clinical trials. Clinically, when we are making a lot of trials, it is very, very useful, and medical research, it is also very important for medical research. It can help in the production of proteins and the drugs in the field of medicine. It is useful for the production of proteins, and it is useful for the production of various types of medicines, drugs in the field of medicines. Then it aids in the stem cell research. Okay, the cloning, animal cloning is a playing important role in stem cell research. Last one, animal cloning could help to save endangered species of organisms. What is the meaning of endangered? Once the population of that particular species was very big, lot of animals were there, but due to some natural changes, okay, climatic changes and various other problems, surrounding problems, the population size is gradually decreasing. That means the death of the individuals will be more, but birth will be less. So the population from millions, it is becoming lakhs. The lakhs, it becomes, the population is becoming thousands. Thousands becomes 100. Now the population is endangered. Any time all the animals will die out. At that time, in the endangered species, we can take the cells and we can make the clones, so identical copies will be produced. Once again the population can be increased, so we can save the endangered species. For that, the cloning is very useful. The cloning is a like plant tissue culture technique, like that it is. Okay, now we are coming to disadvantages of animal cloning. Okay, 8 disadvantages we are going to study. Now, animal and human activists see it as a threat to biodiversity. Now it is a threat to biodiversity. Actually, biodiversity is a natural phenomenon, but in this and all, we are introducing lot of technique. As a result of the introduction of so many techniques, whether biodiversity is affected. Okay, definitely they are telling that side effect will be there, biodiversity will be affected, saying that this alters, which one? The technologies alter the evolutionary sequence will be affected, which will have an impact on populations and their ecosystem. So population growth will be affected, ecosystem is the relationship with all living organisms. Everything will be spoiled. Now there is a first disadvantage. It is only the suspecting, they are suspecting like that. The second one, the process is tedious. Okay, whenever we are dealing with technology, even the previous slide I told, one cell from the udder, one cell, the egg cell from the another mother. But it is not easy. When we are doing, lot of problems will be there. Even small damage in the cell will spoil the entire thing. Once again, they have to start from the beginning. So it is a tedious procedure and very expensive. Now again and again, they have to do the research. For that, lakhs and lakhs, crores and crores of rupees of money will be spent. Then it can cause animals to suffer. Now some of the animals are having their own sufferings. Okay, because whenever we are separating the tissue, animal is suffering. Whenever we are separating the egg from the female reproductive tract, animals will suffer. Then reports show that animal surrogates. Now we have to say, what is a surrogates? Surrogates means a hired mother. One slide before. Yeah. Now once again, surrogate. To explain this, I am coming back to the same sheep cloning. Okay, Dolly, development of Dolly. Now this is the body cell contributor. Second sheep is the egg contributor. But the third one is the mother, actual mother, which is giving the place in the uterus for the fetus to grow. Okay. Now this is the third one is called surrogate mother, surrogate female sheep. Okay. Because it is not having any relationship with this one. It is not having any relationship with this one. It is a separate one, but it is holding a new embryo in it. That is called a surrogate mother. Okay, hired mother. In human being and all, we are telling hired mother, but for animals we are calling it as surrogate animal. Okay. Now we are coming to the surrogate. Reports show that animal surrogates were manifesting adverse outcomes. Okay, we are taking 1/3 animal. It is not having a relationship, but the embryo is implanted into the uterus and the pregnancy is going on. Embryo is growing well, but this mother is suffering or having lot of manifestation of adverse outcomes. Negative problems will be present. Lot of side effects will be present in that animal. And the cloned animals were affected with the diseases. Why the cloned animals are easily affected with the diseases? Now I told the sheep Dolly lived only for 10 days. 11th day it died. Okay, what is the reason? So far the sheep Dolly was kept in the laboratory in a highly protected condition. Immunity is not developed in the body. Immunity is not developed in the body. Once the child, that is the young one, is born, the sheep dolly is born, it was allowed to live in the surrounding factor. Now the animal has a no, the young animal is not having any immunity. Lot of microorganisms attacked this organism. Within 1 week, it developed lot of infectious diseases and finally it died. So that these animals, if you take animals were affected with the diseases and have high mortality rate, death rate. That is, many of the cloned animals are dying very faster than they are living for a long period of time. It might compromise human health, okay, through consumption of cloned animals' meat. Now it is also a threat. Now if cloned animals are there, the cloned animals are giving us egg, the cloned animals are giving us milk, the cloned animals are giving us meat. Suppose we are consuming this egg from the poultry, egg from the cloned, milk from the cloned cow or sheep, okay, or goat. Now what will happen? These product what we are consuming may give some side effect to the human population. It might compromise the human health through consumption of the cloned animals meat. Cloned animals age faster. What is the meaning of age faster? They are becoming old age very faster. Okay, and the lifespan is very less. Okay, instead of living for 6 years, 7 years, 9 years, 10 years, they are living only for 3 years, 2 years, like that. So age faster than the normal animals and are less healthy. They are not very stronger, less healthy than the parent organisms as discovered in sheep dolly. Sheep dolly's parents were stronger, healthy, but sheep dolly was very weak. Cloning can lead to the occurrence of genetic disorders in animals. More than 90% of cloning attempts fail to produce viable offsprings. Now we are telling, from the second sheep, we are taking egg. But when one egg itself, we cannot make success. Failure. Again, they have to take another one egg from another sheep. Third one, fourth one. So to make one dolly, sheep dolly, they wasted 277 eggs. Suppose the 277 eggs were naturally fertilized, 277 young ones would have been formed. But everything is spoiled. Only one sheep dolly was developed, but that was also living only for 10 years. So that is a problem here. More than 90% of the cloning attempts, how much we are attempting? 90%. That is, fails to produce viable offsprings. Right. Ethical issues of biotechnology. Now ethical, the basic principles, natural phenomenon. Now that ethical issues are there. Always whenever we apply any technology, some natural problem will be there. For example, nowadays modernization in the automobiles, in the aircrafts, everything. But everything is a technology. But when we are making this a technology, surrounding is polluted. Now so many other impact will be present. Like that, ethical issues of biotechnology. By applying biotechnology, what are the natural issues are present? We have to see. Many products, what are the products we are getting from the biotechnology, are obtained through biotechnology as per the need based on gene manipulations. We are altering the genes and we are getting the product. People fear that, connected with this point, people fear that there may be unknown consequences by consuming this product. Unknown consequences. What will be the problem? We don't know. Gene manipulation in harmful microbes may deliberately use for biowar. Now this is what? Very important. Now corona, a type of indirect biowar, we can say. Because they started doing the research in viruses, but some viruses, they altered the genes. They made the gene, that is what, viruses to become more and more powerful. So by chance, they are released out. Then immediately the people are affected and it is becoming endemic. Then pandemic. Pandemic means throughout the world, everywhere it is there. Now that is what gene manipulation in viruses is a harmful microbes, that is viruses, may deliberately used for biowar. One country may apply this technology, okay, microorganisms, to leave it in the other country. That is called a biowar, which may lead to epidemic. Epidemic is spreading in that particular area. And environmental catastrophe. The degradation of, catastrophe means degradation. Degradation of the surrounding. Everything will be spoiled. Okay. But in fact, the risk is less and the potential benefits are substantial. But finally, if you take, risk is there. Now we are finding all these things. Risk is there, but the risk is very less, but the product what we are getting is substantial, means more than, yeah, that is enough for us. Now that is beneficial effect is more. So risk is less, beneficial effect is more. So biotechnology is a, that is what applicable we can apply. Okay, we are getting a lot of products. Okay, that's we have completed this topic, very interesting topic. Now this biotechnology is very important topic. After completing +2, if you are opting B.Tech biotechnology in any engineering college, you can do that, but lot of research opportunities and job opportunities are there in foreign countries. Okay, particularly in Western countries, many Western countries, lot of researches are going on, and you can get easily the higher studies. First you have to go, then easily you can get the job, and excellent salary will be given. So you can opt for this biotechnology after completing +2. Thank you. God bless you.